Sir, we are in live, sir.
Okay. Good morning, everyone. I am Mukesh Doble. You must have seen some of my lectures in NPTEL. I'm doing a few courses on NPTEL, and especially this is the biomaterials, medical biomaterials course, which is going on. I think we have completed 4 weeks, and we still have another 4 weeks to go. So I hope you are enjoying the course so far, and you are learning a few new concepts in the area of biomaterials. Biomaterials has become medical, especially the medical biomaterials has become a very important concept, and there is a lot of scope for doing research. There is a lot of scope for industries, startup, especially in India. It's because it's going to be a very big business in the next decade or more, actually. So that way, I hope this course is interesting for you, and you end up learning a few topics on the area of biomaterials. So I thought I will briefly cover what you have done maybe in the past 4 weeks and what is left, and what are the research opportunities in the area of biomaterials. And I also would like to talk about 2 or 3 publications of ours where we have applied these concepts. We have published papers. So I just want to briefly touch upon that, because many of these concepts you learn are used later on. Maybe the analytical tools, maybe the biological assays, maybe conclusions are used in the design of biomaterials. Maybe it's a metallic implant, or maybe it's a wound healing patch, or something like that, actually. So any substance combinations which are synthetic, natural, which are placed inside the human system, treat Okay. Net problem idu. Share screen. Good பண்றேன். ஆமா, I think இதாயிடுச்சு.
So live starts.
Okay, unfortunately, I think we lost the connections. So many journals are there in the area for biomaterials.
Está bien, va. வச்சிருக்கியா வேலையா
இருக்கேன்.
Okay, hello everyone. I think we lost signals for some time. So I hope now it's okay. So I have been talking about some of the topics which must have been covered for you, and some of the topics which we have planned in the next few weeks. Okay. So especially the area of contact between the biomaterial surface and the host system, be it the blood, cells, tissues, organs. That's a very, very important point that needs to be studied. In addition to, of course, its function, its behavior, its mechanical properties or electrical or electronic properties or intended functions, Okay, so there is a lot of scope for collaboration between a biologist and a biomaterial scientist, material scientist, mechanical engineer, and of course you also need a surgeon. Ultimately the product has to go into the market, so obviously the end users, the intended end users should be knowledgeable also. So without a good collaboration with a surgeon or a clinician, maybe cardiologist, maybe a orthopedic surgeon, and the product will never move out of the lab into the uh, actual intended patient actually. So this is a highly interdisciplinary area. We need to keep that point in mind actually, and of course there is a lot of scope for doing research also. Some of the journals in the area of biomaterials, and of course more journals may be coming in, and the way a biologist looks at biomaterial, the way a chemist looks at biomaterial, the way a material scientist or even polymer chemist looks at biomaterial. So we have interdisciplinary journals which are being published from different areas, focusing towards the use or design or application or performance of materials. Okay, so that's why there are many journals that are there now currently, based on your basic area and how you are looking at the material at the end of the day actually. Many companies are there. It's $1 billion business. Even in India, if you look at, it's very huge business, multi-crore business, and it's intended to grow at almost 20%. Okay, very high growth. In the next decade, you are going to see many big companies entering India, and many startups are also going to be in the area of biomaterials. For example, some of these biosensors startups, even IT related companies who are working in this area of materials. So many startups are also entering into, so biomaterials, industrial growth in India is going to be very phenomenal. So hence the research is also going to be very high actually. So as you can see, Netherlands, US, so all over the Europe, US, you can see companies which are in the area of biomaterials. And of course, biomaterials, just like drug needs, the material needs to go through clinical trials and approval from the US, say, Food and Drug Administration, FDA. So it has to go through various phases of clinical trials as well, just like drug, because ultimately it's being placed inside the human body, maybe for a short duration or maybe for a very long time. Suppose someone is having a total knee replacement, the material is going to be there, the knee, artificial knee is going to be there maybe for 10 to 15 years. Stents, cardiovascular stents, it's intended to be there forever. So the requirements are very stringent, and which needs to be going through the process of preclinical trials as well as the clinical trials actually. Some of these where biomaterials are being used, you can see heart valves here. Different types of materials, titanium, cobalt, polyester, polytetrafluoroethylene, polypropylene. So you see, we need expertise from a polymer chemist and of course a material scientist, and then putting them all together. So that's a big challenge, highly interdisciplinary, and putting them all together. Similarly, you can see this is a polylactic acid screw. This is a bioresorbable screw. Otherwise, when you use a metallic screw, it remains inside forever, whereas these screws over a long period of time slowly get bioresorbed. This is used in ligament tear. So again, you need a good expertise in polymer chemistry. You can tailor make the resorbable rate, whether it's going to be completely bioresorbed in 1 year or 2 years or 3 years. So again, there is a lot of scope for research. Again, you can see here orthopedic injury or fracture is a metallic implant, either stainless steel or titanium are being placed. So a lot of scope for material scientists, mechanical engineers to work on design of materials, trying to make it very lightweight, reduce the tensile strength or modulus to bring it to that of the natural bone, prevent corrosions. So many aspects one could look at. Look at this. This is a polymethyl methacrylate beads, where the antibiotic is loaded inside. How do we control the rate of these antibiotics coming out? Can I design biodegradable beads? Because polymethyl methacrylate doesn't degrade, so it remains inside the body. Polymethyl methacrylate is also used in dental implants. So that way it's a very useful, very good material, cheap material also, but it doesn't degrade. So can we design material which are degradable? So that's again, there is a lot of scope for researchers to look at. And these are polyurethane stents. Okay, as you can see here, it's placed inside the kidney and the bladder. So the urine flows through the ureter, especially if there is a blockage because of stones or infections. These are made up of polyurethane, very cheap, very flexible, very lightweight. But uh, after placing it, once the wound is healed, it has to be removed manually. So if there is any polymer which can slowly degrade, then the surgeon does not have to perform another operation. So this is a polylactic acid glycolic acid urethral stent. We have been working on this. So it can degrade. The glycolic acid is very hydrophilic. It degrades. Lactic acid also degrades. So by combining, looking at different ratios of lactic and these two monomers, we can control the rate of degradation. So again, you need a good polymer chemistry expertise to do this. Okay, so can I have a biodegradable stent? It should have the properties of this polyurethane stent currently being used, but it should slowly degrade so the patient does not have to undergo another operation. Okay, so again, polymer chemistry expertise is needed, and of course, the material has to be biocompatible and so on. Actually, these are cardiovascular region. You have shunt, something called shunt placed inside. It lets the blood flow from the aortic branch to pulmonary artery so that it gets oxygenated. Okay, so this is polytetrafluoroethylene. This is a very inert polymer used inside. So it's got a lot of problems. So again, there is plenty of scope for doing research in this area. And of course, anything closer to heart, the performance, the properties, responses have to be very, very stringent. Okay, this is an angiogram picture. You can see the blood flowing here. Sometimes this gets blocked because of so many reasons. I think I'm covering it in the course also. So this is a very good research area. So how do we prevent the blockage of this? These become very stiff over a period of time, and especially, as you know, the heart pumps in a pulsatile manner. So the blood also flows in a pulsatile manner. So can it be made flexible at the same time inert as well as it's strong, so on. So again, a lot of property modifications can be looked at. Okay, so you see, these are some areas which we have been working on, and there is scope for material scientists, polymer chemists, biologists to work together to design material. Some of the materials stays for several years. Some of them will be staying for weeks and months inside the human body. So the requirements are different, designs are different, the locations are different. Some of them, as I showed, is being placed near the urine region where there is salts like calcium, magnesium, salts deposition, bacterial, E. coli, protease, metabolase, like pathogens deposition. Whereas if it is blood contacting region, we have platelets coming into picture. Okay, so proteins coming into picture. So the environments are different, the time durations are different, the requirements are different. So you have different types of design possibilities. These are some knee replacement surgery where they use a polyethylene, cross-linked polyethylene as a spacer between the metallic parts where the ball and socket, as you can see here, are made of metals. So they keep a ultra-high molecular weight polyethylene as a spacer to prevent metal on metal rubbing. Okay, okay. So many things can happen. You can see bacterial can get deposited on some of these material, which can lead to biofilm infection. inflammation, rejection of the material. Okay, so this bacterial contamination or formation of biofilm is a very big issue when the material is placed inside the body. So different types of bacteria can attach depending upon the location. For example, Pseudomonas aeruginosa, it's generally found in the pulmonary region. Okay, that gets deposited. There is Staphylococcus aureus. This is very ubiquitous organism. So when there is an open surgery, these bacteria enter and can start depositing. So the type of modifications to the polymer surface, we need to think differently. Okay, because they have different attachment strength, they have different properties, these organisms, they have different half-life. So we need to design different types of surfaces, different types of drug deliveries, and so on actually. Okay, these are again, you can see some hydrogels, which can be used for wound healing. Okay, they absorb a lot of water. Okay, so they can absorb almost 300% water. Very hydrophilic type of material. There is a lot of work now being done on hydrogels. Okay, these are some bone plates, stainless steel bone plates used inside. Okay, to keep the bone and allow the bone to grow, and you can see some of the screws which keeps the bone plate in place. So when you use different metals, stainless steel, and when you use a different metal for screws, they can lead to corrosion. Okay, different metals can lead to EMF, that means current flow, which can lead to electrochemical corrosion actually. So we have also looked at coating these material with the polylactic acid. Polylactic acid slowly degrades, and we put silver nitrate into this. And as you know, silver nitrate is antibacterial. Okay, so when we coat it, and the polylactic acid slowly degrades, it releases the silver nitrate, so it prevents the bacterial biofilm formation. Okay, so I showed you this bacterial biofilm attachment on these plates, so we can prevent the bacterial biofilm formation. So these are titanium based plates. Titanium has the advantage of two things. Number one, its modulus is less than stainless steel, number one. So it's trying to sort of mimic the actual bone, number one. Okay, and it does not lead to corrosion. Stainless steel has one big disadvantage of getting corroded. Okay, even 316L, it's supposed to be a corrosion resistant stainless steel, but even that can get corroded. Okay, this is a titanium plates, but of course in India still we use a lot of stainless steel because it's cheaper actually. Okay, so biomaterials, metallic materials, okay, organic materials like polymers, biopolymers, a lot of biopolymers, chitosan, chitin, starch, okay, glucon, hydroxy, heptide. All these are biopolymers. And of course, we also use ceramics, carbon, silica, alumina, okay, chromia, zirconia. All these are oxides. So they are ceramic type of biomaterials. Okay, they are inorganic. So there is advantages, disadvantages of each one of them, as you can see. So sometimes they start designing hybrid biomaterials, combining inorganic material with a polymer to get a new property. Okay, that sort of materials are also being looked at actually. So because the duration the material is going to be placed, the location the material is going to be placed, its performance all varies. So you cannot have one solution for all situations. That's why there are a lot of research opportunities for one to go into in the area of this biomaterials, implants, and drug delivery. Okay. For example, inorganic like calcium phosphate or silica, glass, they are very biocompatible, but they do not have mechanical properties like tensile strength. Okay, they just snap, just like chalk piece. If you pull it, it snaps. Okay, so we need to improve that. So maybe they can used as a coating material only. Okay, polymers. are not as strong as metals generally, but they can be made into shapes, different shapes. They can be made into surfaces, could be modified. Okay, so you take biopolymers like glucon or curdlon, right? The strength is very poor, so we need to combine it with metallic, or we need to combine it with inorganic to achieve some of the mechanical properties. Okay, so there is a lot of scope again to look at hybrid biomaterials, combining uh, composites, polymer blends. So again, there is plenty of scope for a research opportunity there. Okay. Um, Okay, so as I said, there are a lot of opportunities for doing research. Let's not go too much into this. Current research topics is one area youngsters who want to enter into the area of biomaterials may be interested to know. So I just made a list of calcium phosphate. Okay, like I said, the inorganics is coming up quite a lot. Tricalcium phosphate, calcium phosphate, silica, okay, bioglass. They are extremely biocompatible. But then only problem is they are also bioresorbable. Okay, that means I don't have to do another surgery like metallic to remove it, but they have some poor mechanical properties. So a lot of work is being looked at improving their properties. Sutureless method for joining blood vessels. So why do you need to have a suture, nylon suture, or even polylactic acid suture? Polylactic acid is biodegradable, but still why have? Can't we just glue it like using araldite like? No? Smart materials, materials which can remember its shape. When the material is made, it has a shape, and then you do something and introduce it, and later on it retains the shape. For example, stents. You want to introduce a stent into a big artery, but the place where you are introducing is very, very small. So you compress the stent and put it inside, and then later on it retains its old shape, like it expands like an umbrella. Okay, that's smart materials, materials which can remember its old shape, okay, or mold properties even. Injectable biomaterials. Okay, so why do a big surgery, open bones, open to do surgery inside, which can lead into bacterial contamination, biofilm formation, infection? Is it possible to just inject a biomaterial? For example, if there is a defect in the bone, maybe cancerous growth, or maybe after an accident, there is a big hole. Can we inject, say, tricalcium phosphate inside? Okay, and it fills up the entire area. And as you know, it's bioresorbable also. So there is now interest in injectable biomaterials. We have also published a paper on injectable biomaterials. Just like take an injectable syringe, make your biomaterial, inorganic material, just inject it. The material should not set when it is being injected, but once it's injected, it should start setting. So the design there is the setting time, its viscosity, how easy it is to, it should not be like water, but at the same, it should go like a toothpaste, but it should set once it has reached its place. So there is a lot of scope to do research in these type of injectable biomaterials. Biodegradable metals, because we have always heard of biodegradable polymers, but why not biodegradable metals? Because metals are have better mechanical properties than polymers. Okay, so now that's why there is some interest on materials like magnesium. As you know, magnesium is bioresorbable, and also magnesium is found in the body, human body, in many cycles of the um, so there is a lot of interest nowadays in magnesium based materials, and of course it's got its own problems. So there is scope for doing research in improving the properties as well as reducing their problems. For example, magnesium, I am going to talk about one of the research which we have done on magnesium based biomaterial with rabbit models. Okay. Microfluidic. Can we make microparticles of polymer using microfluidics? So there is some interest. Many researchers are working in that actually. Neuronal biomaterials, materials that is going to be placed inside the brain. How do we design those type of biomaterials? Because brain, as you know, is very sensitive, so we cannot be making mistake in materials. Biopolymers, as you know, biopolymers or biocompatible. They will not lead to any host toxicity. So there is a lot of interest in biopolymers, but they have many problems because we can't get it in large quantity. Okay, the mechanical properties are extremely poor. Okay, and so on. Actually, balloon catheters. So catheters you insert into very pinholes, but as soon as it reaches its location, it expands like your balloon. So there is this direct sensing electrodes. Okay, electrodes which are placed inside, maybe for left ventricle assisted devices or certain pacemakers. Electrodes design. So I think electrical engineers or even electronics engineers can find interest in that. Biosensors. Nowadays, there are a lot of interest in live sensing of the various human properties. Okay, can I sense continuously the blood glucose level, or can I sense the pressure and other properties continuously, monitor it, especially for patients who are very prone to various diseases which can lead to sudden attack. So can we have live biosensors which transmit the data to their surgeons or clinicians, or to other instruments which will automatically correct those problems? So again, electronic engineers can play a very important role. Aesthetic appearance, of course, this is going to be a billion-dollar business. Facial surgeries, maxillofacial surgeries, okay, all these cosmetic surgeries for various body parts which are destroyed or damaged because of due to birth defects or after a trauma. So that's again a very big business. I think that will keep growing and growing and growing. It will never be stopping at all. The laser processing of biomaterials. How do you prepare, process? 3D printing. That's already started happening over the past decade. Printing, metallic 3D printing, polymeric 3D printing, bioprinting. So can we introduce cells during printing? The cells then later on start growing. We can have a printable material to suit each person instead of having a standard, just like shoes, 7, 8, 9, and so on shoes. Sometimes we feel 7 is too tight and 8 is too loose. So why not have shoes which are printed for each of our foot? Okay, similarly, why not have if somebody is having a stent placement for an angioplasty, why not make a stent to suit his or her arterial dimensions? Okay, so I think why not have maxillofacial features printed for each individual? Why have a standard size or shape inserted in the face, and the face gets completely modified? So I think that's going to be a very exciting area in the next decade, the 3D printing of various body parts, actually. Tissue engineering, of course, is another area, and design of scaffolds externally and then placed inside 3D scaffolds. Blood-brain barrier penetration, because there is a barrier which prevents blood flowing into the brain. It allows only oxygen, nutrients. It prevents bacteria and even drugs to penetrate. If I want to give a CNS drug, neural drugs for neuronal defects, Parkinson's disease or Alzheimer's, how do we allow the drug to enter? Because this barrier is supposed to prevent anything entering. Apart from nutrients, oxygen, how do we allow it to enter? So there is again, can we design biomaterials which will do this job? So we can have the drug made to be carried through that biomaterial into the central nervous system for blood-brain barrier penetration. Then bone graft substitute for large defects. Small defects is very simple. Small defects means up to 2 centimeters. But if there are defects which are larger after an accident or after a corrective surgery, can we have substitutes? Multiple drugs delivery. Can I deliver one drug and after a few hours or days deliver another drug? Can we have an antibiotic and an anti-inflammatory delivered? Can we have an antibiotic and a growth factor delivered? Can we have different growth factors delivered at various periods of time, which could be hours or which could be even be days? So again, that's a big area in the area of drug delivery. Not just one single drug in one shot, but doing this type of intelligent drug delivery. Hydrogels, aerogels, as I said, they are very hydrophilic and they take in a lot of water. So that is another good area to work on actually. Okay, then imaging, different types of imaging, live imaging, accelerate platelet aggregation, nano topography on metals. This is called biomimetics. Okay, can we have designs of surfaces which look similar to some of the features of, say, shark skin, and so on. Then prevent biofilms in implant defect, understanding the mechanism. Ultimately, scientists would like to know why this particular event happens. Why does platelet start activating and start aggregating when I place a particular biomaterial? So finally, understanding the mechanism is very, very important for scientists. Okay, stress-related mechanisms, immuno-related responses, inflammatory responses. Why does the implants fail? Okay, that's another big area. Many biomaterials fail after a few hours, after a few days, and after a few months. So why do they fail? That's if you go to FDA site and then look at biomaterial failure, and they give list of failure which has happened and why the material is recalled. So you can see poor sterilization, failure of electronic controls because biosensors and some of the left valve and left ventricle assisted device or even pacemakers have electronics. So they fail. Catheters, sutures, mechanical failure, tensile strength or wear strength or flexural strength. Environmental stress cracking, bimetallic corrosion. If you have two metals, always there will be a problem of corrosion. Okay, and especially in orthopedic region, you'll have a screw, you'll have a metal, sometimes two metals connected, okay, cobalt or chromium or nickel, and corrosion, stress corrosion. Okay, mechanical failures, stress shielding, biodegradation. You don't want material to degrade, but there are regions where it will start degrading some polymers. Absence of osteo integration. That means two materials don't integrate properly with the tissue. They come out loosened. It's called loosening of material. Infection, biofilm formation. So these are some of the reasons for failure of device. So again, scientists, engineers, and researchers are looking into this. Okay, to prevent this. Okay, and this is from the FDA site, as I said, inadequate sterilization. So this is a very big area for orthopedic surgery tool. Steel rust, that means corrosion. Ventilators with defective component, infant resuscitators with assembly error, guide wires with flaking coating. Guide wires are used especially if somebody is going to have angioplasty. Okay, the stent is taken inside. And tracheal tube, that King's, unclear labeling on left ventricle assisted device. So this is what FDA, they give the recent medical devices failure actually. And as I said, I would like to show some of my couple of papers, depending upon how much time we have, where we have used some of these techniques. Okay, this is a very interesting research which we have done, where you have used magnesium alloy as an implant into a rabbit long bone segmental defect. And currently titanium implants are placed, especially for long bone defect, which is bit defect or breakage, which are greater than 2 centimeters. But as you know, titanium or stainless steel do not biodegrade, so they remain in the body forever. So there is some interest in using magnesium alloys. Okay, AZ31 is nothing but aluminum, zinc uh, of 3:1, 3% and 1% in magnesium to make these implants. And as you know, magnesium is bioresorbable. So we wanted to look at whether we could place these magnesium cages in the defect of the rabbit. Magnesium resorbs very fast. So the problem is it will completely disappear before the bone heals. So we want to slow down this bioresorption. So what we did was we coated this with poly and caprolactone. Okay, so the biodegradation of magnesium is slowed down. Okay. If you use titanium metals or stainless steel, the problem is they remain inside the body forever. So that's why there is some interest in bioresorbable metals like magnesium. And as you can see here, cardiac surgeon is an orthopedic surgeon. So without a collaboration with the surgeon, nothing could be done. And of course, if you want to do animal studies, we also need help from a veterinarian. So we are the research scientist. We have a collaboration with an orthopedic surgeon. We have a collaboration with someone who is running an animal lab. And we also have a collaboration with another scientist who is a microbiologist from Sri Chitra Tirunal Institute of Higher Learning, Trivandrum. So the whole project is a collaboration between, as you can see, different institutes and different expertise. So without these type of collaborations, biomaterial research will never see move from the lab to the patient. Okay, let's go to the problem. I'll just show you some of these pictures. So this is magnesium cage. This is approximately 2 centimeter long magnesium cage. So when we keep it inside the body of an animal, it will bioresorb very fast. It will not be sufficient for the bone to heal. So what we did was we coated these with the polycaprolactone and hydroxyapatite. As you know, the bone contains a lot of hydroxyapatite, so it's biocompatible. So there won't be any adverse reaction or cytotoxicity. And polycaprolactone is also a polymer approved by FDA, and it degrades slowly. So we are trying to retard the degradation of this magnesium implant. Okay. Okay, a lot of characterization has to be done, like Fourier transform in spectroscopy and X-ray crystallography to measure the X-ray plane. Fourier transform infrared spectroscopy gives you an idea about the various chemical groups that are present on the surface. Okay, so this is how the surgery is done. As you can see, the defect in a rabbit, the magnesium coated cages. You can see this is the uncoated cage. Then this is the coated cage, and after the surgery, here is being sutured inside. Okay, so after about 4 weeks, the cages are removed, the uncoated ones and the coated ones. Okay, and then the performance of the bone growth is studied, and the changes in these implants that are studied. Okay, so you can see some X-ray pictures. First day, second week, fourth week, sixth week, 8 weeks, okay, and so on. So you can see the product which we have developed, which is a magnesium cage with coating of polycaprolactone and hydroxyapatite, allows the bone to heal very well. You can see the bone growth, the tissue growth, as well as the bone growth. We can study the bone growth looking at the bone density. Okay. When we have the uncoated material, the bone growth, as you can see, is not good. You can still see the gap here, but here you can see it's healed very well in 8 weeks. Okay, so then we use CT image, micro CT, computer tomography. Then it gives you an artificial coloring to see how the bone growth has taken place, how the bone density also could be calculated. The bone density, these are called micro CT. Okay. Then we studied how they helped bone volume improvement, total porosity, okay, then the bone thickness. All these could be studied from these CT images. Okay, and then we do see that there is, and then of course histopathology studies of how the surrounding tissues have grown, whether there are any inflammatory responses, and so on actually. So we are able to prove that the design which we have created using a magnesium alloy coated with the polycaprolactone and hydroxyapatite is able to nicely allow the femoral defect, long bone segmental defect in rabbit in 8 weeks. The bone is able to heal very well, and the bone density also is improved. There is no inflammatory response. Okay, the surrounding tissues also have grown very well in 8 weeks, and it has been shown very well in a rabbit model. So that's a very interesting problem. As you can see, it uses a lot of mechanical design. It uses a lot of analytical tools. It uses a lot of orthopedic surgeon expertise to perform the surgery, and then a lot of data analysis. Another interesting problem I would like to show to you. Here we are looking at silver-loaded nanofibrous curdlan. Curdlan is a biopolymer. It's non-cytotoxic, biocompatible biopolymer. So we have loaded silver in fiber of curdlan as a wound healing application for diabetic rats. Yes, so we have studied on diabetic rats. Okay, so curdlan is a biopolymer. Okay, so it's biocompatible, and we use silver nitrate, as you know, which is antibacterial. So we incorporate that into these curdlan mats. We prepare these curdlan mats using electrospinning technique. So fibers of curdlan comes out with silver nitrate. Then we look at diabetic wound healing in rats. That is the whole surgery study here. Sorry. Study here. So again, you see animal studies are being shown. So it's not just enough to do lab studies, because lab studies and going into animal is a very big step. Okay, and then later on, of course, we need to go into human, but without animal and studies, just doing in the lab alone has no meaning at all. Okay, so these are curdlan fibers you can see here. Okay, nice fibers which are prepared by electrospinning technique. So in electrospinning, the polymer solution is injected into a needle and made to come out in like a jet. So there is a voltage applied on the plate. So there is an voltage at the needle and voltage applied at collecting plate, we call it. So these jets of polymer solution comes and hits the collector plate as a fiber. Okay, so you get these fiber mats. So we can incorporate drugs into these fibers. We can incorporate anything. We have looked at even RNA delivery, siRNA delivery. We have looked at drugs delivery, so many things. But here we are putting silver nitrate. So, and look at these beautiful fibers. They're all in nanoscale fibers, because the scale bar is 2 micron, and you can see the fibers or nanometer scale, and different amounts of silver nitrate can be studied, and silver nitrate is antibacterial, and of course a lot of characterization. So wound healing, the first problem in wound healing is infection. So the first 4 or 5 days we need to prevent the infection building up, because infection can lead to a lot of problems of infection and inflammation and so on actually. So a lot of in vitro studies are done initially in the lab. So we look at how the bacterial interaction with the silver nitrate impregnated in curdlan. all done in vitro initially, and then later on we see how this, whether the material is biocompatible, because it's going to be in contact with the wound, so it has to be biocompatible. It should not lead to any inflammation. So we see how the cells are growing, how the cells are viable. These are some pictures of that. And then we also do something called cell migration assay. This is done in vitro. What we do is we allow the cells to grow, and then we make a scratch artificially with the glass rod, and then over a period of time we see how the cells are migrating and closing these the scratch created. You can see them, cells are growing nicely. This is called an in vitro study of cell migration. This is an in vitro study. In vivo, we have to do animal studies, but in vitro is useful to tell whether the cells are migrating. You can see this, okay, after 24 hours, nicely. Okay, this is control where we don't do anything, and this is with our product of wound healing patch. Nicely, it's growing up, right? So we have studied the infection preventing ability in vitro with bacteria. We have looked at how biocompatible this product is with cell line, and we see how the cells migrate and close the gap which is produced artificially. All these are done in vitro. So we are happy with that. So what we do is we go into the animal studies. Okay, so when we go into animal studies, yeah, these are wounds created on the animal surface, diabetic animal. That means the blood glucose levels are very high, 250, okay, deciliter per deciliter. Then we put in our patch. Okay, you can see here, this is our patch, and within 14 days it's completely healed. Whereas this is the control, 14 days, that means control is no treatment, the wound is still there. As you can see here, this is completely healed, and this is a commercial product. After 14 days still the wound is there. So this is a commercial product. This is our product, which has a Kurlan fibers, nanofibers, with the silver nitrate impregnated. The silver nitrate does the job of antibacterial activity. Kurlan is known to have immunomodulatory property, which is approved by FDA. So the combination of antibacterial and immunomodulatory properties help the wound to heal here. As you can see very well, wound healing that's taken place in 14 days when compared to this control disease, and this is a commercial product. Again, you see a lot of expertise in biology, in vitro studies, and of course support from the veterinarian to perform these type of wound healing study in animals. So now this product, of course, if one wants to go take it further, and data on animal studies, that is preclinical studies, is already available actually. So this is a biopolymer. So biopolymer, especially in the area of wound healing, we don't require much of mechanical strength. So it's okay. Otherwise, if you want to use it for applications where you need to, it needs to look at mechanical load bearing application, we need to use some other synthetic polymer. We need to prepare a blend of these biopolymer with the synthetic polymer, whereas in wound healing application, it's not going to face much of tensile issues or compressive strength, so it's okay. So you can see the nanofibers which we have prepared has done a wonderful job of retaining the drug and then releasing it over a period of time to arrive at a very good wound healing within 14 days. Okay, so that's a very interesting, another interesting problem. Finally, let me show you a third problem where we have used sorry. Okay, here we have used it a polymer for drug delivery and in the area of tuberculosis, actually. See, the Mycobacterium tuberculosis enters macrophages and then they reside inside. So, designing drugs which will enter these macrophages, that is infected macrophages, and killing the mycobacterium is a big challenge, actually. So, we need to have a delivery system which can enter this. Okay, so this is the challenge here. So we looked at a polymer called curdlan. Curdlan is a biopolymer. It is produced by bacteria. Even many plants, cereals contain curdlan. They have immunomodulatory properties. They are biocompatible. Curdlan is not soluble in water. So what we did was we converted it into curdlan sulfate. So we are able to prepare a soluble product, and then we incorporated drugs into it. Curdlan has certain property of identifying receptors in the macrophage. Macrophage has a receptor called dectin-1, and this curdlan has a beta configuration, and this beta configuration can recognize the dectin-1 receptor, so it can very selectively target the macrophages. That's the beauty of this particular polymer which we have selected. So we didn't select any biopolymer or any synthetic polymer, but we selected a polymer curdlan which has this beta linkage configuration, which can recognize the dectin-1 receptor found in macrophages to enter into the macrophages. So we have a drug, an antibiotic, which is encapsulated into this, and so the biopolymer can specifically go and bind to the dectin-1 receptor found in the macrophages. Okay, so it can selectively target these infected macrophages, because targeting infected macrophages is the biggest challenge in the area of tuberculosis, because the mycobacterium enters and becomes dormant, okay, for a long time. Okay, this is a very interesting research problem which we looked at. Okay, so what we do is the curdlan is a biocompatible material produced here. We produced it using a bacteria, and then later we converted that into a curdlan sulfate to make it water soluble. Okay, I just want to show you. Yeah, and then we encapsulated a drug called rifampicin. Rifampicin is a drug which is used in tuberculosis, but it's not very efficient in entering these infected macrophages. So we encapsulated this rifampicin in curdlan sulfate. And as I said, curdlan has this beta configuration or beta linkage, which can go identify the dectin-1 receptor found in macrophages. Okay, so you can see some scanning electron pictures of these nanoparticles which we have prepared. Okay, then we look at how the drug gets released over a period of time. Okay, so these are those studies, like, and then we tried to fit these into various mathematical model drug release, and then we see whether the polymer drug which system which we prepared is biocompatible, whether it kills the cells, okay, or whether it's biocompatible. That's very, very important, because it's being placed inside the body. Okay, so this is another picture of fluorescent images. You can see microscopic images of these macrophages. Okay, the macrophages incubated with the curdlan sulfate nanoparticles with the rifampicin drug. Okay, you can see this, and then we see how the nanoparticles enter into these infected macrophages. Okay. Okay, then we see how the drug enters, especially when we have only the drug alone. The entry of the drug into the macrophages is very poor, but whereas when we have with the encapsulated into the nanoparticle, the entry is much, much higher when compared to the free drug alone. So we are able to design or get a system which has better chances of entering into the infected macrophages. Okay, so this type of design can be used for targeting certain types of cells which have this dectin-1 receptor. So we can introduce drugs into it, or we can introduce a certain RNA or any other growth factors or signaling molecules into this. So this is a very interesting system which we try to design to achieve these type of properties. Okay. Okay, so we looked at many things. And especially I had, because the biomaterials and drug delivery is a highly research focused area. So I looked at I showed you some of these current research topics that one could enter into. And I also showed you a few papers of ours where we have used our knowledge of biomaterials and tissue engineering for implant as well as for drug delivery applications actually. Okay, so if you want to take biomaterials as one of your research topics in your future career, I think there is a lot of scope for that. So let me stop with this. I hope you have been benefited in the past 4 weeks, and I hope you enjoy the course. It will expose you to some of the fundamental concepts in biomaterials, and of course, the area of research, the scope is infinite. You need a support of mechanical engineers, polymer chemists. You need support from material scientists, and with your expertise as biologist, I think we could look at various types of implants, tissue engineering, drug delivery, and so on. Actually, so best of luck for the remaining duration of the course, and wish you all the luck in your final NPTEL exam as well. So hopefully I will have another session where I would like to show you, demonstrate the instruments which we have in the area of biomaterials. Unfortunately, you might not be able to come, be present physically, but I will try to show the operations of the instruments through this online mode. Okay, thanks for being part of this. I hope you benefited, and if you have any doubts, you can write to me. Of course, I have given you my email ID. You can note down the email ID again. So if you have any doubts for your future research as students, okay, so you can definitely write to me, and I will like to help you out in the doubts. Actually. Okay, thanks for your time. Thank you very much, and I hope you enjoyed this session. Okay, thank you.
Hello, sir.